Dr. Peter McCaffrey. He's founder and chief technology officer of a company called Pragma Biosciences, based in the Bay Area in San Francisco. So Peter got his medical training from Hopkins, and also did his residency at Mass General, and he's a certified clinical pathologist.
Yeah.
And his talk is Metagenomic Enzymes, a window into evolution for next generation pharmaceuticals, essentially secondary metabolite encoding genes from bacteria. That's encoded for pharmaceuticals targeting different diseases, including infectious disease.
Yeah.
Peter.
Perfect. So I'll talk on the title a bit. I want to make a comment that's kind of interesting, both futuristic and anachronistic at the same time. So we talk about next generation pharmaceuticals as sort of an opportunity made available to us by understanding metagenomic enzymes and engineering those enzymes. This is, of course, something that we've seen grow a lot recently with synthetic biology. But it's worth noting, this is the oldest method of drug discovery, actually. This is the evolutionary scale discovery. Enzymes have built virtually every organic material in the universe that we know of, save very recent history. So it's more like a new window into the past. So why do we care about enzymes? Well, a few reasons that might be obvious to people in this group. Evolution is sort of the ultimate design algorithm. It built us, it built everything we have, basically, everything that lives on our planet and likely beyond. And it operates in a major way through optimization of functional units, as we know. And many of those functional units are enzymes. Not the only thing evolution exerts pressure on, but it does exert pressure on enzymes to do very amazing things, both in singleton and collaboratively. There's a lot of embedded knowledge in the footprints of what evolution has done to enzymes, and the ability to recapitulate those enzymes and modify those enzymes. Bringing our sort of capabilities of design onto a scaffold that has already solved some more fundamental problems of discovery. So we look at enzymes at Pragma Biosciences, of course. And by the way, we were formerly called Vast Biome. You may have seen us present at a conference like this recently. We were actually fellows of the Texas Medical Center way back, biodesign program, some half-decade ago, before founding the company and spinning out and moving to the Bay Area. So we were in Houston once upon a time, both Kareem and myself. So what do we really focus on? Enzymes are everywhere, obviously. You can find enzymes in all genomes, basically. But we focus on a particular kind of enzyme superstructure, you can say. They're called biosynthetic gene clusters. You may be familiar with these. And basically, these are when you look at the genome of an organism, particularly bacteria in our case, although you can see it in fungi, you can see it in many other things as well. You see enzymes that occur adjacent to one another inside of a genome. They're basically collaborating by virtue of co-occurrence to make a biosynthetic pathway. So why would you care about that? Well, you care about that because you get typically more specialized, what they call secondary metabolites. So if you hunt for biosynthetic gene clusters, what you essentially are hunting for are the biosynthetic recipes to make exotic chemistry that nature has already refined in organisms. Now, maybe it didn't refine them under the target objective you have in mind, but it's important to note that there's lots of constraining parameters apart from target effect that might prevent you from de novo discovery of something like this. These include things like fundamental enzyme kinetics, compatibility of substrate, thermodynamic stability, and so forth. There's a big lift done for you by layering your design on top of a biosynthetic gene cluster that has already paved the way to make something relevant. Okay. What do I mean by relevant? What would be an example of something that is relevant under my conception? Well, us at Pragma, we consider lots of different natural products as relevant and interesting. You essentially have limitless possibility looking in the natural space, but it's not very useful to just say we look at everything. You can't really do that effectively. So where do you lean in? What do you care about? Well, it's worth to note that there are fascinating areas of natural product space all over. But one area that is particularly intriguing is what are called ribosomally synthesized, post-translationally modified peptides, economically RIPs, we can say, from here on out. And why would we care about RIPs? Well, RIPs are protein products protein products have some really fundamentally interesting properties. I would name these properties broadly as designability. If you think kind of broadly over the space of machine learning, computational design, and analysis of structures that you want to make, it may be something that we all realize that these tend to work quite well on sequence analysis, sequence substitution, and sequence to structure prediction. This is not equally relevant in all cases, of course. If trying to do this with pure chemistry, that chain of reasoning does not necessarily hold as well as when maybe doing it with sequence design. This is why we see things like antibody design or RNA therapeutic design or RNA vaccine design be very feasible and successful in certain ways, especially when guided by machine learning. Turns out, for the same reason we're good at doing this with language, we're good at sequence to sequence and sequence to meaning and sequence to structure, basically sequence to endpoint representation. And there is a grammar of proteins, just like there's a grammar of language. So you might say, okay, why care about metagenomic enzymes and proteins? What's unique about this space? Why not just make a peptide or an antibody? Well, again, you go back to nature and what nature has decided to do with some of these things. So what I show you here is an example of a RIP of acute interest to us at Pragma, known as a lasso peptide. So a lasso peptide is one of the many RIPs that can exist, has its own ontologies and subclasses and divisions, as everything does. But you can generally consider a RIP, or a lasso peptide, to be a RIP that is a protein folded into this like lariat, like a knot structure, as you see here. And why would we care about the knot? We care about the knot because, of course, the knot is actually very stable to degradation whilst still having externally exposed epitopes and being derived from a peptide that is modified, cyclized, tied off, and cleaved. And so when you think about how you can leverage this for design, if you consider the biosynthetic pathway of a lasso peptide, you have what's called the core peptide, which is a linear initial substrate, usually printed within the BGC. Then you have a parsimonious set of enzymes, let's say 3 or 4 tailoring enzymes that do things like cyclize, dehydrate, and cleave. What's also fascinating about this is that the global sequence of the core is not fixed for it to be compatible with the enzymes. So you see how nature has kind of given you some optionality in design space, where you say there are regions of the core that must be conserved, there are regions that do not have to be conserved, for them to be fed into the same enzymes to produce a lasso. In other words, I have the ability to hijack this 4-enzyme cascade, but swap the core peptide for the production of alternative outputs that both start at a local space of molecules that nature has found, but let me imbue my target effects and interest into that if I want to optimize a molecule to bind or behave a certain way. So our technology platform basically looks at this problem in the context of what we think might address a patient phenotype, either because we believe we can make a target, a protein target a protein of interest, or because we find that BGC broadly correlated in a phenotype of interest. So our interest is broadly the gut immune axis. We like to hunt for things in the space of human metagenome data, where we identify BGCs, those that are conserved in patients who survive, obviously, and patients who exhibit phenotypes of interest to us, such as response to immunotherapy, response to IBD treatment, for example. We say what kind of natural products exist in these patients, what kind of epitopes are there, and can we locate and refine these? So can we find them in computational space? Can we perform cell-free expression to recapitulate the enzymes? Can we literally reproduce those products in a plate-based controlled liquid handling format? And then can we screen them for effect? So this is kind of what it looks like when you do this enzyme BGC hunting against a human data set. This you see right here is a real reflection of a data set that's created off of an aggregate of published studies looking at, for example, human response to immuno-oncology therapy. These are patients who mainly got anti-PD-1, anti-PD-L1 therapies who had various response rates 6 months out, scored by RECIST criteria, who had variable gut microbiomes in the setting of a consistent therapeutic program they were being submitted to. And we asked ourselves the question, can we hunt in areas of BGCs where we think they're conserved in responders, they're impermanent in non-responders, it might lead us into a place to hunt and to design. And so what we do with the data analysis that goes into this is we take each patient, their gut microbiome, and convert it into like a bag of BGCs, if you will, just a set of BGCs. And we consider across the whole population which BGCs are conserved, shared between people, and shared between people and phenotype. And then we cluster these by similarity, sequence similarity to the BGCs, and correlation with phenotype. And you essentially pick areas of intense clustering. You say, if I see a knot, if you will, in this cluster, I'm therefore seeing examples where I have lots of enzyme BGCs that are similar and they're shared by different people, and yet they were conserved in responders and not conserved in non-responders. My assumption is they're making a class of relevance to this axis. And so we take this BGC template, recapitulate the biosynthesis of the product, and then derivatize by alteration of the DNA that leads to the creation of the enzymes, or in this case, the core peptide. So what an example of this look like? So the platform leveraging these enzymes is you basically say, I want to make a protein of interest. I want to make this, and this enzyme that comprise a BGC. I don't want to culture microbes in the process of making that because they're often fastidious and that is difficult. So how can I bring this more to an engineering capacity? Well, I can say, I want to make this protein. I design an oligo. I ligate that DNA oligo in between cis-regulatory elements, a promoter, a terminator, et cetera. I do cell-free expression on batch. I pluck successfully expressed enzymes, I repool them, and I feed their substrate. I subject this to mass spec depending on the category of product I expect to produce. Could be a peptide, could be a polyketide, could be a terpenoid. Of course, an interest of us is mainly in peptides and lactopeptides, but we look at a broad portfolio of potential products. And then we screen these, and we screen these basically on single well attributes we call things like cytokine induction against a control PBMC cell set in a plate, or single cell data, sometimes single cell sequencing, primarily things like FACS analysis. And we say, can I recapitulate a phenotype that these BGC product is inducing over human immune cells? So if I do this, then I've basically discovered enzymes that have broad correlation that I can make, that I can refine, that I can reconstitute, that I can recapitulate an effect on. So you capture kind of a product you want to make and a biosynthetic route to that product, oftentimes which involves intersecting with chemistry that is exotic and difficult to reproduce in vitro. Again, going back to the case of a lactopeptide, which is our current area of primary focus, and our library is comprised half of lactopeptides at the moment. These are things you can't produce in vitro. You can't produce this exotic dehydrated lariat knot formation in vitro. You have to do this through enzymatic control. Another example of this would be highly stereoselective products. You see these in some NRPSs or PKSs where you have many, many chiral centers. And of course, your in vitro efficiency of production is exceedingly low, because you're basically going to fractionate your yield every time you have a chiral center. But evolution has solved this problem that we grapple with in making very predictable stereoselective synthetic antibodies that can overcome this kind of thing and create preferential chirally selective products. And so really, you don't have to rely exclusively on nature as the only way to find things. But if you search in the unrestricted space of natural products, it's basically hopelessly broad to find stuff. And so you start in a place that nature has teed up as relevant, then you apply synthetic biology to override that and derivatize locally. We use this database we conventionally call lattice, which really reflects the way we represent all of these products as a graph. Many-to-many relationships between genes, their BGCs, their pathways, their products, their phenotype. And through this connection, potentially their target, if it is known. And it's grown essentially over the last few years, in 2018, 2020, 2021 now, to be a pretty dense graph that essentially lets us do two particular types of analytical methods on this, looking at enzymes, enzyme groupings. One being probabilistic graphs, so simply what is the probability if I have a certain set of enzymes that it will tie to a phenotype, things like Bayesian graphs, PGMs, if you're familiar with this kind of thing. And of course, embeddings, and especially in sort of the post-LLM world, embeddings and powerful linguistic embeddings and embeddings applied to proteins, things like ProtTrans, things like ProtGPT2, really allow for powerful comparisons in ways that are very machine understandable and easy to translate into statistically important enzymes, enzyme groups, and enzyme phenotype associations. But of course, since you have a graph of labeled entities, you can do interesting ad hoc queries into this. Here's an example of zooming in on an area of the graph and saying things like, this is really where you're essentially asking evolution to answer the question for you in part, how I make something. You're saying, I want to find a BGC, for example, that it contains an enzyme I know I want to use, like let's say a cyclase that I'm interested in. I want to find all the other associated enzymes with that BGC, and from those, the ones whose reaction is understood, and from those, the one whose substrate might be understood, and from those, the ones where I can procure that maybe off of sigma, for example. Letting me kind of look at what we know about what nature has decided to do in pairing enzymes, substrates, reactions, and series of those things, to find sort of smaller biosynthetic groupings I can recapitulate and work with. And so this query basically says, of a BGC of interest, I know that these 3 enzymes are commonly conserved. They work together in this way. I can use them in isolation by feeding this substrate and falling through the product, and I can kind of excavate this from evolutionary space and work with it in a more controlled environment. And so, in summary, drug discovery in this space, what sort of evolutionary enzymology allows us to do is be able to consider this as a multi-parameter optimization, if you will. So you look at a space and you say, I have a phenotype I want to approach, or maybe even a target I want to approach. I want to look at phenotype-based screens, things like gut microbiome data for associated BGCs, associated taxa. That's one sort of question. But then in that space, I want to consider, from what I know about enzymes, their kinetics, their biosynthetic role, their potential biological effect, their product class, and tee me up an enzyme that I can work with, a BGC that I can work with, I can recapitulate, and I can test in vitro. So I'm going to be kind of quick and leave some space for questions at the end, and sort of there we kind of have as an overview.
And great to learn about these lasso peptides. Never heard of them before. So are they being used, or are you using them to look at their potential for maybe disrupting protein-protein interactions and with in vivo?
They do exist in narrow use cases currently. Things like microsin J25, for example, is a lasso that is used. But on the whole, they're rarely seen fully translated into practice. And so you consider, why would you care about them? Well, they're peptides, they're designable as peptides. But the most curious thing about lassos is their stability. The lariat offers a stability that is unique to this space in that they are resistant to enzymatic degradation. Meaning that you might gain advantages of something like antibody-based epitope grafting and design without the trade-off of lacking PO availability, possibly. So we're early in our investigations here. But an attraction we have to these is, can you have a degradation-resistant payload that might be more versatile in route of administration? Because typically, you'd have to make that trade-off very much up front. But you might not have to. And again, this would have been difficult to invent if it wasn't for the fact that nature has kind of already done this, because gut-resident microbes make this, and they have to survive in this context and produce these. And so we kind of look into that as a way to approach the problem differently. But yeah.
So back in the 90s, there was a huge kind of focus on natural products and kind of people kind of fishing out broths from different places and kind of throwing those broths on cells and looking for phenotypes.
Yeah.
Have you ever gone back to some of that old data just to see if you can find a BGC and kind of knowing that there's something there that's already got a phenotype?
Yes, actually. So some of the interesting correlations that have popped out, like in the earlier work we did at the very beginning, we looked at things like of broad taxonomic associations. If you consider a taxonomic lysate, like you know the bugs that are conserved in response, if you had a lysate of those bugs, would you see that response again? What mechanism would it have? We consider this analysis both looking at literature that focused on taxa and dissecting lysates of taxa, and BGCs that we thought could explain that. One particular example of something that would come out of this would be zwitterionic capsular polysaccharides, or polysaccharide A, which have in fact been explored by others as a potential immunoregulatory, even a cancer adjuvant, but have other kind of class-based problems, like the fact that they're polysaccharides. So sometimes it's a modality problem. And of course, in the microbiome space, you have lots of things that are like membranous polysaccharides, peptidoglycans. Things are just fundamentally difficult to productize in a reliable way and maybe hard to design. So it's really a series of trade-offs to try to get to something that is both naturally occurring and active, and yet also something you feel like you can crystallize. I think a lot of what we've seen in the space that has come out as being like clearly correlated, that's an example. Non-BGC products like butyrate is another example, are either not exotic enough or not of the right kind of compound class to consider like translatable or commercializable or designable. And so, we are of course optimistic this will be surfaced, but this is kind of the challenge. They're cool, but like they don't make it all the way to like this would be a program.